Hello friends, you all are welcome in my channel College Botany Online Free Learning, and you are studying the enzymes with Yuvraj. So you are going to learning the botany paper 8, paper code is D.Sc. D14, and title of paper is Plant Metabolism. It consists two units. Unit number 1, enzymes and nitrogen metabolism. It will deal by me. And unit second, respiration, seed dormancy and germination. It will deal by Dr. Magdum sir. So let's first discuss regarding the enzymes. So see here, the topic enzymes comprise the syllabi. The first point is introduction, the classification and nomenclature of enzymes, structure and properties of enzymes, mechanism of enzyme action. Two mechanisms we will study: lock and key hypothesis and induced feed hypothesis. Next, factors affecting on enzyme activity, temperature and pH. And last point, the enzyme inhibition. So basically we are knowing that there are two fundamental conditions for the life. The first one is the living organism must be able to replicate, and second is the organism must be able to catalyze the chemical reactions efficiently and selectively. These chemical reactions are catalyzed by a special remarkable proteins, and they are called as enzymes. So enzymes are the most important constituent of biological world and have made life possible on the surface of earth. The word enzyme is derived from Greek language. In means in and zyme means Easter, or it is also meaning as a leaven. So something is there which is living in the yeast cell that was considered as the enzymes. So basically enzymes are a catalyst. So you are looking here, the substrate get bind on the enzyme, substrate enzyme complex form, and finally the product get formed, enzyme get liberated. In this way enzymes are taking part in the biological reactions. So the enzymes are a biocatalyst. So catalysts are the substances which increase the rate of reactions without changing their cells, are called as a catalyst. So enzymes are the protein, they inhale there in the cell and accelerate the rate of reactions, therefore they are called as a biocatalyst. So enzymes have the extraordinary catalytic property, and they are highly specific to the substrate. So enzymes accelerate the rate of biochemical reactions without changing their nature. They are taking part in the biological reactions, but At the final, they escape from the biological reactions and impart in other biological reactions. So, are the enzymes protein? Yes, the enzymes are proteins. So, proteins, a simple proteins, a conjugated proteins, or complex form of proteins are worked as enzymes. So proteins are nothing but a chain of amino acids. They exhibit the primary structure, secondary structure, tertiary structure, and highly modified quaternary structures. So the arrangement of the primary, secondary, tertiary, and quaternary protein structures formed the active sites. So active sites are nothing but the place where substrate get bind and reaction, biochemical reaction get accelerated. So by definition, the enzymes can be defined as a complex chemical protein produced in a living cell which catalyzes a biochemical reaction at a very low concentrations. The scientist Meyerbeck defined the enzyme are simple or compound protein acting as specific catalyst or the organic substances capable of catalyzing chemical reaction in a living system are called as enzymes. The discovery of enzyme was takes place by the Greek and Roman people. They produce the wine, they use the grape for the fermentation, and during this process the yeast is used, and in that yeast some kind of substances might be present, and due to that the fermentation takes place. So that substances are nothing but the enzymes. So fermentation of sugar into the alcohol is the first reaction which stressed the birth of enzyme to the all humankind. The scientist Louis Pasteur has recognized the potentiality of yeast cell to ferment sugar into the alcohol. So Louis Pasteur used a long neck flask for the fermentation process. The scientist Frederick Kuhne firstly coined the term enzyme. This is very important. Scientist Kuhne firstly coined the term enzyme. He said that any chemical substance generally called ferment. So J.B. Sumner firstly isolated and purified the crystal of enzyme urease. So these are the crystals, and this discovery is remarkable in the enzymology. The pepsin and trypsin are the digestive enzymes. This was firstly recognized by the scientist John Northrop and Kunitz. So this pepsin are secreted by the gastric glands and involved in digestion of protein. So there are many enzymes discovered by the scientists. They find their structure, chemical nature, and mode of actions. So all these enzymes and their activities now we are going to learning in this topic. And basically we are knowing that the all enzymes are protein, but all proteins are not enzyme. Thank you. Thank you very much.